There are two general systems that are going to sort protein in the cell to their final destination. The first one is called co-translational import, and the second one is called post-translational import. So remember, every single protein has a place in a cell, a place that it's going to live and function. And all of those proteins have to get delivered to wherever they're going. But this delivery, this sorting, happens much sooner than you would think. It's not that, oh, everything happens to the protein first and then it gets sent to its destination. Instead, for many proteins, this is a sorting process that occurs while translation is still going on. That's what co-translation means. Now remember, I have a star next to co-translational import because this is the system, the process that we're going to spend the most time talking about. We'll say just a few things about post-translational import, which is the system that sends or sorts a protein to its destination after translation has occurred. Now, all of this sorting, whether it's co-translational or post-translational, all of this sorting depends upon something called a signal or a signal sequence. It turns out that polypeptides have a few amino acids on them that are going to be used by the cell almost like an address on an envelope. They are the signal that tells the cell where this protein needs to go, where it doesn't need to be sorted to, where is its final destination going to be. So, any protein that is ultimately destined to live in the rough endoplasmic reticulum, or the smooth endoplasmic reticulum, or the Golgi apparatus, or a transport vesicle, or a lysosome, or the plasma membrane, any protein whose final destination is going to be in one of these places, is going to be sorted using co-translational import. Now, any protein whose final destination is to be inside the cytoplasm, inside the nucleus, in the mitochondria, in the chloroplast, or in a peroxisome, any protein who is destined to live and function in one of these locations, that protein will be sorted through post-translational import. Now take a look at this very complex diagram. We're going to look at this diagram several times. Right now, I just want to orient you to what you're looking at. Up at the top is a very basic looking image that shows us how ribosomes interact with an mRNA, shown here as a brown string. The ribosome, shown in this blue-green color, interacts with that mRNA, travels down the length of the mRNA through translocation, remember, and produces a polypeptide, a string of amino acids. All right, we're comfortable with this. We've talked about this in detail. Now look at the bottom of the diagram. On the left-hand side, we're looking at the process called co-translational import. And as you can see in the picture, here's what we just looked at, the ribosome and the mRNA and translation going on. And here are the organelles that are going to step in and become involved. The blue represents the rough endoplasmic reticulum. The purple represents the Golgi apparatus. You can see transport vesicles labeled here as purple secretory vesicles, and then these blue lysosomes, and then this plasma membrane. Remember, these are all the final destinations for proteins that are sorted through co-translational import. On the other side of the diagram, again, you can see the ribosome and the mRNA up at the top, but then the other thing you see are the destinations involved in post-translational import. The nucleus, the mitochondria, the chloroplast, the peroxisome, or just the plain old cytoplasm. All right, these are the two processes that are involved in sorting, and we're going to focus on this one. So let's get started. The first thing to remember is that ribosomes inside of a cell, a eukaryotic cell specifically, can either be located on the surface of the rough endoplasmic reticulum or free floating in the cytoplasm. You can find ribosomes just in the cytoplasm of any cell. And you can also find ribosomes sitting on the membrane surface of the rough endoplasmic reticulum. Co-translational import is this process that sorts proteins to their final destination, but begins by pulling the growing polypeptide that's being created through translation, pulling it down into the rough endoplasmic reticulum, into the lumen or the cavity or the space inside all of those flattened sacs of rough endoplasmic reticulum. Co-translational import gets its name because the first part of it is happening while the ribosome is out there translating the mRNA. As the polypeptide is being produced during translation, the polypeptide is going to be imported or pulled into the lumen of the rough endoplasmic reticulum. Now, how does this happen? Well, the ribosomes, which normally, again, sit on the membrane of the rough endoplasmic reticulum, they're found there as well as out in the cytoplasm. The ribosomes are going to become bound to that membrane temporarily while this happens. During translation, the new polypeptide, which is being produced through translation, is going to get pulled in to the rough endoplasmic reticulum so that as translation keeps going, instead of the polypeptide being produced out in the cytoplasm, it's actually going to be fed into the rough endoplasmic reticulum. That's how it gets in. Eventually, translation is going to be done, and the polypeptide will be released into the rough endoplasmic reticulum. And then all that processing goes on that I mentioned a minute ago. Cleaving and splicing and glycosylation and so on. Now, when all of that is done, that processed protein, a new protein, because it will have been folded as well, is going to get packaged up into a transport vesicle and it will exit the rough endoplasmic reticulum. But it's not done. It also now needs to move into the Golgi apparatus where it will be processed further. It'll get packed again into a transport vesicle, and then it will be sent to its final destination. And for co-translational import, remember that means it's either going to the rough or the smooth ER, it's going to the Golgi, it's going to live in a lysosome, it's going to live on a plasma membrane. Somewhere in that list is going to be its final destination. Now, how does this happen? How does a polypeptide get pulled in to the lumen of the rough endoplasmic reticulum? How does that happen? What is it that signals to the cell that this polypeptide, as it's being translated, needs to be pulled down into the rough endoplasmic reticulum? Why doesn't it just get produced in the cytoplasm where the ribosome lives? A couple of scientists named Blobel and Sabatini figured this out back in the early 1970s, and they referred to their theory as the signal hypothesis. Blobel and Sabatini figured out that every polypeptide that gets produced through translation has a short sequence of amino acids at one end that functions as a signal. Again, almost like the address on an envelope. The first several amino acids on that polypeptide are not going to become part of the final protein. Their job is just an address on an envelope. Their job is just to act like a signal. So the signal hypothesis tells us that on some polypeptides, that sequence, that signal, the first few amino acids of a polypeptide, is the address for the rough endoplasmic reticulum. And the way it works is that ER signal sequence is about 15 to 30 amino acids long, and that signal sequence is going to help guide that polypeptide as it's being translated over to and into the rough endoplasmic reticulum. So one of the hardest parts of wrapping your head around co-translational import and post-translational import is the idea that for every protein that gets produced in a cell, part of its original structure, part of its original sequence of amino acids, that means part of its original genetic code, includes a few amino acids whose only job it is to get that polypeptide where it needs to go in the cell. So even back here in the DNA, in the gene, in the protein coding part of a gene, some of that code is not for what will become the final protein, but simply what will become an amino acid signal that will direct the young, immature polypeptide to its final place in the cell. So you have to imagine that there's a gene in a piece of DNA, and that gene is coding for a protein that's going to be sorted through co-translational import. So we are located right now in your mind's eye in the cytoplasm, but near the rough endoplasmic reticulum. The ribosome is on the surface of the rough endoplasmic reticulum, but it's not attached to it yet. An mRNA has come out of the nucleus, and the ribosome has jumped on it, and it has started translating it. So it has moved down to the first codon, the start codon, and a tRNA has brought in the first amino acid, which is always methionine. And then it translocates to the next codon, and translation gets underway. That ribosome is going to translate the first 15 to 20 codons. And now we have a string of amino acids, 15 to 20 of them, that is an ER signal sequence. Okay? Now, that ER signal sequence you might think is going to now bind to the rough endoplasmic reticulum. That would make sense, I think. But that's actually not what happens. The ER signal sequence doesn't actually contact the rough endoplasmic reticulum. Instead, there's a middleman involved. There's a piece of protein called a signal recognition particle, or an SRP. It's actually protein and some RNA. The signal recognition particle is going to recognize and bind to the rough endoplasmic reticulum. It's going to bind in the area of the membrane on the RER that contains this big structure called a translocon. Now, what is a translocon? It's a combination of four things, and I've got them listed here. It's going to have a signal recognition particle receptor. It's going to have a ribosome receptor. It's going to have a pore, a passageway that's capable of opening up and sending something across the RER membrane. And finally, what's called signal peptidase. This is an enzyme, and as it sits inside of the translocon, it's inactive, at least temporarily. This enzyme is capable of removing the ER signal sequence from a polypeptide once its job is done. On this slide, we can see another complex diagram. This one is all about the signal sequence and the translocon. Now, there are 6 panels on this diagram. We're going to take a few minutes to go through all 6. But for right now, just start in the upper left-hand corner with me. We are sitting in the cytoplasm in this drawing. The brown ribbon represents a messenger RNA. It's just come out of the nucleus. It's ready for translation. The blue-green molecule represents a ribosome that has attached to the mRNA and has started translation. Now, what you're seeing faintly, I know, right here in yellow is the polypeptide that's being produced by the ribosome, the amino acid chain that's being produced by the ribosome. You'll notice that it's relatively short here, and the very first part of it has a blue color. This little blue area, this represents the first, again, 15, 20 amino acids. This is the ER signal sequence. Now, over here, there's a little brown molecule labeled SRP, signal recognition particle. It's made out of protein and RNA. And this particle is going to come along and it's going to bind to the blue area on the polypeptide, the signal, the ER signal sequence. Now, over here in the next panel, we've moved everything from the cytosol to a connection with the rough endoplasmic reticulum. That's what you're looking at in blue. This whole region right here is the translocon. So what you're looking at in dark blue is the membrane of the rough endoplasmic reticulum, and in light blue is the inside cavity or lumen of the rough endoplasmic reticulum. Now in this panel, what has happened is the signal recognition particle, after it binds to the signal sequence, it sort of drags it over and the SRP binds to its receptor in the translocon. So this brown SRP, this signal recognition particle, has now brought the signal sequence and the translocon together. It acted like a middleman, and it brought the whole ribosome assembly in close contact with the membrane of the rough endoplasmic reticulum. Let's look at the rest of the translocon in this image. The purple region, that's going to be the pore that I mentioned a minute ago. This dark sort of turquoise color structure, this is the ribosome receptor. So when the SRP comes in and binds to its receptor, the ribosome will also bind to its receptor, and the whole thing becomes much more stable. The final part of the translocon is this brown region over here. This is that inactive enzyme, that signal peptidase enzyme. It's inactive right now, but it will play an important role later on. So let's walk through the steps so far. We said that so far, a ribosome started translating some mRNA. The first 15 to 20 amino acids were produced, and a signal recognition particle bound to them. Then that signal recognition particle bound to its receptor on this structure called the translocon on the rough endoplasmic reticulum membrane. And when it did that, the ribosome also bound to the translocon in the area of the ribosome receptor. So we got this big piece of machinery set up, and we got the ribosome and the mRNA in contact with it. Now remember, the translocon has a pore in it, a channel, and that pore is going to be used to translocate or move the growing polypeptide into the rough endoplasmic reticulum. Now, hopefully it makes sense to you that all of this so far sounds like it requires energy. It sounds like a lot of work, doesn't it? All this binding and moving and attaching. And sure enough, we do need some energy for this to occur. That energy is not going to come from ATP being hydrolyzed, but instead GTP is going to get hydrolyzed. The GTP is going to have its outermost phosphate group removed so that we get GDP, that phosphate group, and a little bit of energy. What that energy is going to do is help the next step happen. It's going to help transfer the signal sequence, again on the end of the polypeptide, into the pore. And the pore will broadly open up so that the polypeptide can keep coming down through the pore into the rough endoplasmic reticulum as it's being translated. The pore is going to open up so that we can move this long chain of amino acids down into the rough ER. Now at that point in this process, the signal recognition particle is going to get released. It's done its job. It brought the signal sequence in contact with the pore. It's done its work. It can be released now. Let's go back to this drawing and take a look at this. So remember, we stopped here. We had the signal recognition particle binding to the signal sequence on the polypeptide, the blue, and also binding to its own receptor, shown in this beige color. We had the ribosome too, binding to its receptor in turquoise. Now we're going to hydrolyze some GTP to get a little bit of energy for what happens next. The next thing that's going to happen is the pore is going to open. The signal sequence, the blue part, is going to enter the pore. It's actually going to bind with the inner wall of the pore. And then, as we move to this panel, we're going to set our signal recognition particle free. It's done its job. Look at how it's all set up now. We have the ribosome translating the mRNA. We're still doing that. A polypeptide is being produced. A chain of amino acids is being produced. The very first part of that chain is now bound to the wall of this pore or this tunnel. So as the chain of amino acids gets longer and longer and longer and longer, it's just going to pass down into the lumen of the ER. That's what they're showing us here. The ribosome keeps moving down the mRNA. It keeps adding one amino acid at a time. The blue strand here is just getting longer and longer and longer. It's being fed down through that pore into the lumen of the ER. So as that polypeptide continues to be translated, it's going to be fed down through the pore into the lumen of the rough endoplasmic reticulum. Remember, eventually the ribosome is going to come to the stop codon on the mRNA, and translation is going to stop. That means that the polypeptide will be released from the ribosome. The rest of it, that last little bit, will fall through the pore into the RER. And now we need the enzyme, the signal peptidase enzyme. Remember, that's the part of that big translocon structure that functions as an enzyme. And what it's going to do is break off the signal sequence from the rest of the polypeptide. It's going to cleave or break the bond between the amino acids of the signal sequence and the amino acids of the rest of the polypeptide. So that polypeptide can fall down into the lumen of the rough endoplasmic reticulum. It will be completely released at that point from the translocon. The pore is going to close. Outside of the pore, the ribosome will detach from the translocon. Translation is over, so it will disassociate from the mRNA, and we're done. And what has happened is the cell has, during translation, brought this brand new polypeptide into the lumen of the rough endoplasmic reticulum. And if we go back to our drawing, that's what we can see. Remember, we stopped here. The ribosome is out in the cytoplasm. It's now bound to the membrane of the rough endoplasmic reticulum, but it's still out in the cytoplasm. It's translating this brown mRNA. It's producing this blue polypeptide that is coming down through this pore in the translocon into the rough endoplasmic reticulum cavity or lumen. At some point, the ribosome is going to hit a stop codon. And we know from our review of translation that when that happens, when it's finished translating, it's going to let go of the polypeptide. But remember, there's still an issue here because the signal sequence is still bound to the pore. So that's when this part of the translocon gets activated. It's going to cut at the junction between the signal sequence and the rest of the polypeptide. It's going to make a cut. And this happens. The polypeptide is now set free, set loose from the ER signal sequence that was at its very beginning. The pore is going to close. The ribosome is going to be released from the membrane of the rough ER. It's going to break apart because it's done with its work right now. The mRNA will go off and be degraded. And now we have this brand new polypeptide sitting in the lumen of the rough endoplasmic reticulum. Now, just one more word about co-translational import here. That particular polypeptide that we just watched as it was made through translation, that polypeptide had an ER signal sequence at one end. It had a group of 15 to 20 amino acids that was the address of the rough endoplasmic reticulum. There are other proteins that are sorted through co-translational import that aren't going to live in the rough ER. Remember the list? Rough ER, smooth ER, Golgi apparatus, lysosome, plasma membrane, etc. there are several destinations in the cell that proteins get sorted to through co-translational import. So depending upon where those things are going to end up, there are going to be other parts of the polypeptide that serve as signals. There are going to be other regions, segments of amino acids in the polypeptide that serve as signal sequences. So that when that polypeptide is all done, passing through the rough ER, and it gets all processed and folded up, there will be a little place on that folded up protein that is an address. It's going to be a signal that says, okay, now that I'm all folded up, just make sure that you send me to this final destination. As soon as I'm ready to go, make sure I get sorted to this final destination, either the Golgi apparatus or a transport vesicle or the plasma membrane or wherever else it's going to go in the cell.